Good morning, ladies and gentlemen. My name is Professor Titilayo Bamidele, and I'll be taking you chemistry, structures, and functions of lipids, which is a part or a section of general biochemistry, BCH211. So at the end of this lecture, you as students should be able to, number one, define lipids. You should be able to tell me what a lipid is. Number two, you must be able to classify lipids based on their structures and functions. Number three, you must be able to write the structures of examples of each of the classes of the lipids that we would have discussed. Number four, you must be able to name some common lipids and their functions. Number five, you must be able to enumerate the physical and chemical properties and biological functions of each of the families of these lipids. And finally, you must be able to describe the reactions of fatty acids, such as esterification, hydrolysis, saponification, and hydrogenation. Now, let's start by defining lipids. What are lipids? Lipids are organic substances that are relatively insoluble in water, but soluble in organic solvents, such as chloroform, ether, and benzene. When you mix lipids with water, it does not dissolve inside the water. For example, in your house, you have oil, your palm oil, your vegetable oil. If you drop it inside water, you will see that it does not dissolve properly inside water. So they are organic substances which are relatively insoluble in water. And they are insoluble because they have larger non-polar alkyl groups. They have non-polar alkyl groups. These alkyl groups makes them insoluble or poorly soluble in water. So as the size of the alkyl group increases in an organic compound, the water solubility of the compound decreases. So the increase in size of alkyl group will contribute to the insolubility of the lipid inside water. So the higher the size, or the bigger the size of the alkyl group, then the less soluble the lipid becomes. So as a result of their non-polar character, lipids typically dissolve more readily in non-polar solvents. So because of that non-polar character, lipids will generally dissolve in non-polar solvents such as acetone, ether, chloroform, and benzene. All these are non-polar solvents, and lipids will dissolve in it more than it will dissolve in water. Now, these are the general biochemical functions of lipids. All right, these are the functions a lipid performs. Number one is storage form. A lipid is a storage form of energy. The lipids are generally used in the generation of energy. So they are storage form of energy. Most lipids are stored. Even your carbohydrates does not get stored as much as lipids inside the body. So they are stored in the adipose tissues, and when carbohydrates are not available, these lipids are made use of to generate energy. Number two, they are precursors of many steroid hormones and vitamin D. Lipids are used in the synthesis of steroid hormones, and these steroid hormones are chemical messengers in our bodies, that transmits information from one part of the body to another. And also in the synthesis of vitamin D, we all know the importance of vitamin D in the body. Vitamin D is very, very important for the bone, mineralization of the bone, and so on and so forth. Then they serve as chemical messengers. For example, you have the inositol triphosphate, IP3. IP3 are messengers. They carry information from one part of the, in the cell. So the lipids can serve, the inositol triphosphate is an example of a lipid, and it serves as chemical messenger, that is, they transmit message in the cell. Then they act as electric insulator in neurons. Lipids insulate the neurons. The neurons are where you have that's where you have transmission of nerve impulses. So lipids insulate the neurons. Then fat serves as surfactants by reducing surface tensions. Okay. Then also lipids form prostaglandins. Some lipids form prostaglandins, and these prostaglandins are very, very important in inflammation. They serve, to regulate inflammation. Lipids also helps in absorption of fat-soluble vitamins. The fat-soluble vitamins are vitamin A, vitamin D, vitamin E, and vitamin K. So lipids, they are not soluble inside water. These vitamins are not soluble inside water, so they need a lipid environment. That's why they are called fat-soluble vitamins. So for their absorption, lipid is very, very important. Also, lipid serves as padding for internal organs. They serve to cushion the internal organs so that they will be preserved from injuries. So they pad the internal organs. And finally, their function, in the function of lipids, they are structural components of cell membrane. The cell membranes are made up of lipids, especially the phospholipids. So lipid is very, very important in the formation of cell membranes. So these are the biochemical functions of lipids. So what I've explained there is what I have in bullet points here. They are a major way of storing chemical energy and carbon atoms in the body. Fats also surround and insulate vital body organs, providing protection from mechanical shock and preventing excessive loss of heat energy. Also, phospholipids, glycolipids, and cholesterol are the basic components of the cell membranes. Then several cholesterol derivative functions as chemical messengers in the body. That is, they are converted, the hormones, steroid hormones especially, are synthesized from cholesterol, and they serve as chemical messengers in the body. Also, they help in the absorption of fat-soluble vitamins. I've described it. I've told you the fat-soluble vitamins are vitamins A, vitamins D, vitamins E, and vitamin K. These are fat-soluble vitamins. They need the lipids to be able to get absorbed in the body. Then they function as electrical insulators in neurons and serves as surfactants by reducing surface tensions. These are the functions of lipids. Now, we have been able to look at the functions of lipids. How are these lipids classified? How are they classified? What we call lipids, they are classified. But how are they classified? They are classified majorly into three classes. Number one, we have the simple lipids. Number two, the complex lipids. Number three, the derived lipids. Simple lipids, complex lipids, and the derived lipids. These complex lipids are also known as compound lipids. They are also known as compound lipids. Now let's take them one after the other. For simple lipids are basically the esters of fatty acids with various alcohols. When you have esters of fatty acids with alcohols, they are called simple lipids, and they are of two types, namely fat and oils is one, then the waxes. The alcohol in fat and oil is glycerol, while the alcohol is they are long chain alcohol that are esterified to fatty acids to form waxes. Now, the fat and oil, they are also known as neutral fats. They are also known as neutral fats. And these are esters of fatty acids with glycerol. Glycerol is the alcohol here. This, if you look at this blue box, that is the glycerol backbone. And these are fats, fatty acids, esterified to the backbone, to the glycerol backbone. So we have 3 fatty acids esterified at position 1, position 2, and position 3. Now this is called triacyl glycerol. This compound that we have here is triacyl glycerol. Tri means you have 3. 1, 2, 3. Tri. Then fatty acid, they are known as acyl. Triacyl, then glycerol. So it can be in the first and second position. That will be a diacyl. If the fatty acid is esterified to one position, it's called monoacyl glycerol. So this here is a neutral fat and is the esters of fatty acid with glycerol. Now this is the glycerol. It's an alcohol, right? And this carbon is carbon 1, this is carbon 2, and this is carbon 3. Then we have an ester bonding. An ester bonding normally occurs between a carboxylic acid and an alcohol, all right, where you have the alcohol donating a hydrogen and from the carboxylic acid and OH to form water. So the bond that is formed between an alcohol, between a fatty acid and an alcohol, is known as an ester bond, ester link. So this is a monoacyl glycerol, that is one fatty acid is esterified at position number 1 to the glycerol. It's a monoacyl glycerol. The second one is a diacyl glycerol. You can see that you have 2 fatty acids being esterified at position 1 and at position 2. It's called a diacyl glycerol. And this carbon 2 here is a chiral center. A chiral center is a carbon that is attached to 4 different compounds. The compounds that they are attached to are different from each other, so it's called a chiral center. If you see, it's attached to CH2 here, it's attached to this one, it's attached to this compound which is different from what is attached to here, and is attached to another compound which is entirely different. So the 4 attachments around this carbon are totally different. So this carbon is a chiral center. And the last one is triacyl glycerol. Just like I've told you, when you have 3 fatty acid in ester bond with glycerol at position 1, position 2, and position 3, you call it triacyl glycerol, which is called fat. Now, function of triacyl glycerol. Triglycerides, it could either be called triacyl glycerols or triglycerides. Triglycerides serve as a source of energy for the body. All right, they are the source of energy. They are stored in the body and they serve as source of energy for the body. Triglycerides also help to insulate the body and protect vital organs. If you look at where fats are stored, especially the belly, those are where we have adipose tissues. You discover that these triglycerides, they get deposited in your belly and also around some organs. So what they do is they also, aside the fact that they are sources of energy, they also protect the vital organs against mechanical or any injury whatsoever. So the 2 major functions of triglycerides are that they serve as source of energy and they also insulate the body and protect the vital organs. For waxes are different from the neutral fats because of the alcohol. So waxes are esters of fatty acid. Fa is fatty acid with higher molecular weight monohydric alcohols. Instead of cholesterol, for fat it is cholesterol, but for waxes it is monohydric alcohols. So examples of waxes are lanolin, your beeswax, your whale's palm oil. They are examples of waxes. Now, this is how a wax is formed. It is also an ester linkage. This is the palmitic acid, which is a fatty acid, and then you have the myricyl alcohol or triacontanol, and the alcohol here is a long chain alcohol, C30CH61OH. So myricyl alcohol is the alcohol here. This is beeswax. Now, palmitic acid plus myricyl alcohol or this myricyl alcohol is also known as triacontanol. Now, the ester linkage is formed. I've told you that ester linkage is formed between a carboxylic acid and an alcohol. So this is the ester linkage that is formed. So this is an example of waxes. So waxes are simple lipids made up of fatty acid chains esterified to long chain alcohols, and the alcohol may contain 12 to 32 carbon. So it's actually esterified to a long chain alcohol. Now, function of waxes. Number one, they are strongly hydrophobic in nature. So this allows them to function as water repellents on the leaves of some plants, on feathers of birds, and on the corticals of certain insects. So they serve as water repellents. You can see when water drops on feathers of birds, once they are shaken off, it goes off because they are water repellent. So they serve as water repellent. They also serve as energy storage substances in plankton. Plankton are microscopic aquatic plants and animals, and also in higher members of the aquatic food chain. So they can also serve as energy storage and also as water repellent. Now we have learned about simple lipids. Simple lipids are basically esters of fatty acids and alcohol, and the alcohol could be either glycerol or long chain alcohol, just like we have seen in beeswax that the alcohol is myricyl alcohol. Now, for complex lipids or compound lipids, I've told you they can also be called compound lipids. They are esters of fatty acid with alcohol, and they can contain additional groups, prosthetic groups. And these complex lipids are subclassified according to the type of prosthetic group. We have about 3 of them, 3 complex lipids. Number 1 are the phospholipids. Number 2 are the glycolipids. Number 3 are the lipoproteins. Now, phospholipids generally are fatty acids plus alcohol plus phosphoric acid, and they frequently have nitrogen containing bases. All right, aside the fatty acid, aside the alcohol, aside the phosphoric acid, they contain nitrogen containing bases. So this is a schematic diagram of a phospholipid. This is the glycerol, which is the alcohol, which is the backbone. Then you have the fatty acid being esterified to the glycerol backbone. Then you have a phosphate group, all right, and this phosphate group can also be linked to this nitrogen containing bases. The phospholipids may be classified on the basis of the type of alcohol that is present. Phospholipids can be classified as glycerophospholipids and can be classified as sphingolipids. Now, for glycerophospholipids, the alcohol here is glycerol. You can see it is glycerol. While for sphingolipids, the alcohol is sphingosine. It's sphingosine. And for glycerophospholipids, you have two fatty acids being esterified. Then you have the phosphate group, and then the alcohol. The alcohol are also known as the nitrogen bases. They are also alcohol. It can be choline, it can be ethanolamine, it can be inositol. We'll see in the next slide. Now, for the sphingolipids, you have the sphingosine as the alcohol. Then you have a fatty acid esterified. Then you have the phosphate and the choline, which is the nitrogen containing base or alcohol that it contains here. So for phospholipids, you can either have them as glycerophospholipids that contains glycerol as a backbone, or sphingolipids that contains sphingosine as the backbone. So examples of glycerophospholipids, we have said the alcohol is glycerol, are phosphatidylcholine, phosphatidyl ethanolamine, phosphatidyl serine, phosphatidyl inositol, plasmalogens, and cardiolipins. For for sphingolipids, sphingospholipids, the alcohol is sphingosine, and then an example is sphingomyelin. Now, these are the structures, examples and structures of phospholipids. The first one is phosphatidic acid. Phosphatidic acid has only phosphate group, attached to the diacylglycerol, so to say. So just the the phosphate group alone. So this is phosphatidic acid. Number 2 is phosphatidylcholine. Is also known as lecithin, or contains choline, all right, attached to the phosphate group, which is attached to the diglycerol. The third one is cephalin, which is phosphatidyletanolamine, just like the choline. Ethanolamine is attached here. Then phosphatidylinositol, where you have the inositol group attached to the phosphate. Then you have phosphatidylserine. This is the serine group attached to the phosphate. And then you have the plasmalogen. All right, plasmalogen, instead of an ester linkage here, you remember we have an ester linkage here. What you have here is an ether link. All right, it's an ether link. That is the difference between plasmalogen and phosphatidylethanolamine. Where you have ester link here, what you have is an ether link here. So that's plasmalogen. It's also an example of phospholipid, and it also contains ethanolamine. So for sphingolipids, we have said that the sphingosine is the alcohol. You can see the sphingosine, that is the alcohol of sphingolipids. Then you have a fatty acid that is esterified. Then you have phosphate and choline. So this is an example of sphingolipids, sphingomyelin. This is the fatty acid residue esterified. This is the sphingosine, which is the alcohol backbone to which this fatty acid is esterified. So this whole one, this whole compound here is the sphingosine. Then this is the phosphate, and this is the choline group compound. This is choline. So the whole of it are known as sphingomyelin. They are known as sphingomyelin. Now, functions of phospholipids. Number 1, in association with proteins, phospholipids form the structural components of membranes, and they regulate membrane permeability. Their major function is that they, with proteins, they form the structural components of membranes. All membranes are made up of phospholipid, whether it is the glycerophospholipid or the sphingophospholipid. They form the structural components of membrane, thereby regulating membrane permeability. All cells, cell membranes are they are not permeable like that. That is, things don't flow into them, whether it's ions, whether it's their molecules, does not just flow inside the cell, so they are not permeable, and it's because of the lipids that are present on the membrane. The lipids regulate the membrane permeability. So we said, we normally say that cells are selectively permeable. They allow some things, but they don't allow many things to enter. So they are selectively permeable. Then phospholipids also participate in the absorption of fat from the intestine. It is also essential for the synthesis of different lipoproteins, and thus participate in the transport of lipids. So phospholipids participate in the transport of lipids, since they are involved in the synthesis, or they are essential for the synthesis of lipoproteins. The lipoproteins transport lipids around the body. Then accumulation of fat in the liver can be prevented by phospholipids. Hence, they are regarded as lipotropic factors. So they prevent accumulation of fat around the liver, or prevent fatty liver. Number 5 now is arachidonic acid, which is an unsaturated fatty acid that is liberated from phospholipids, serves as a precursor for the synthesis of eicosanoids. For example, the prostaglandins, the prostacyclins, the thromboxanes. All these are required in inflammation, prevention of inflammation. So, and this arachidonic acid are synthesized from phospholipids, and also all these are gotten from arachidonic acid. So lipids is important in the synthesis of prostaglandins. Still continues, they participate in the reverse cholesterol transport, and thus they help in the removal of cholesterol from the body. So phospholipids remove cholesterol from the body. Number 7 now, phospholipid acts as surfactants, that is the lower surface tension. Then you have cephalins, which is an important group of phospholipids. They participate in blood clotting. All right, and then phospholipids, which example phosphatidylinosytol, is involved in signal transmission across membranes. I've told you that there are some messages that are passed from one place to another, all right, and they come in form of signal. So a phosphatidylinosytol helps in transmitting these signals across membranes. The second type of complex or compound lipids are the glycolipids. The glycolipids are slightly different from the phospholipids. The glycolipid contains fatty acid, contains alcohol, which is also sphingosine, but it contains carbohydrate with nitrogen base. All right, Instead, phospholipid does not contain carbohydrate. Glycolipid, from this word glyco means it has carbohydrate. Glycolipids, so it contains carbohydrate with the nitrogen base. And this particular glycolipid, they do not contain phosphate groups. That's the difference between phospholipids and glycolipids. The carbohydrate which glycolipid contains, and the phosphate group which phospholipid contains, but it is not present in glycolipids. Examples are the cerebrocytes and the gangliosides. Now these are examples of glycolipids. These are just schematic diagrams. You have the ceramide. The ceramide contains just the fatty acid and the sphingosine, that is ceramide. For the cerebrocyte, cerebrocyte has the the fatty acid, the sphingosine, and a sugar. Then you have the sphingosine. The sphingosine is the alcohol to which you have these groups attached. Then you have the sulfatide. The sulfatide has a sulfite group attached to the sugar. Then you have the fatty acid, and then you have the sphingosine, that's sulfatide. Then you have the ganglioside. Ganglioside has complex sugars attached, all right, and you have the fatty acid and also the sphingosine. All these are examples of glycolipids: the ceramide, the cerebrocyte, the sulfatide, and the ganglioside. You will observe that there is no phosphate in all these, all right. That's the difference between them and the phospholipid. They still have sphingosine, just like the sphingospholipids, and they also have fatty acid esterified to the sphingosine alcohol, but they do not contain phosphate. They contain sugar. You can see cerebrocyte has a sugar, ganglioside has a complex sugar, whereas sulfatide also has a sugar and a sulfite group attached. These are the structure of the glycolipids. This is a structure of the ceramide. Okay, it's a ceramide. This is the sphingosine molecule, whereas this is the fatty acid. All right. Then you have the ceramide. This is the sphingosine, all right, without the fatty acid. The ceramide has the fatty acid. Okay, you can see the fatty acid. Then you have a choline group. This is sphingosine molecule. Then you have the ceramide, which has the fatty acid. Okay, so another example is the cerebrocyte. This is the glucose molecule attached at the carbon at this junction, all right. Then you have the fatty acid, and these are the sphingosine molecules. The sphingosine molecule, which is glucose is attached, and you have the fatty acid. The ganglioside has a complex oligosaccharide. That means you have about four sugar molecules attached, all right. So this is the sphingosine. The sphingosine is the pink, whereas you have the fatty acid. Then you have the complex oligosaccharide, the ganglioside and the cerebrocyte. The function of glycolipids, number one, is that they maintain the stability of the cell membrane. Maintains the stability of the cell membrane. Okay, they join with the lipids. They are with the lipids on the cell membrane, all right, and they provide stability of the cell membrane. Number two, They facilitate cellular recognition, which is crucial to the immune response. Their presence on the membrane facilitates cellular recognition. They could recognize self from non-self. So, and this is very, very important in immune response, whereas you recognize pathogenic substances which are not self, and the immune will respond by destroying the pathogenic organism. Also, they allow cells to connect one another to form tissues. They form their presence on the membrane allows them to recognize other cells that belongs to a particular tissue, and then they connect to one another to form tissue. So they are very, very important for self or for recognition, whether cells that look like them so that they can aggregate together to form tissues, or cells that don't look like whatever is present in the body, and therefore to the kind of there is a launch of the immune response, to counteract such cell. The lipoproteins, just like we have said, they are lipid with protein. From the word lipoproteins, means lipid and proteins. So you have lipid with protein as prosthetic group. Examples of these lipoproteins are the chylomicrons, the very low density lipoproteins, the low density lipoproteins, the high density lipoproteins. All these carry lipids in the body. They carry lipids around in the body. So they are very, very important in the transportation of proteins in the body. The last class of lipids are the derived lipids. Derived lipids are the lipids that are gotten from hydrolysis of complex and simple lipids. So examples of derived lipids are fatty acid that you can get when you hydrolyze both compound lipids and the simple lipids, especially the fats. Also cholesterol. They are a separate class of lipids which they are not gotten from hydrolysis of both the simple and the compound lipids, but they are also lipids in their own right. They are classified as lipids. And from cholesterol you can get steroids, and then vitamin D. All these are linked. These three are linked: steroids and vitamin D. All these are examples of derived lipids. These are structure of cholesterol. These are the steroid skeleton. You have A, B, C, and D rings. All right, and number one, this is number 1 carbon here. Number 2, number 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So you have 17 carbon on these rings. Then this is cholesterol. This is the structure of cholesterol as a methyl group attached at carbon 10, then another carbon 13, and then it has this chain attached at carbon 17. The structure of ergosterol and stigmasterol. All these are examples of steroids. All right, the example of steroids. So vitamin D, or calciferol, is the general name for a collection of steroid-like substances which include vitamin D2, ergocalciferol, and vitamin D3, which is cholecalciferol. and they are found only in animal sources. All these are found in animal sources. Vitamin D is fat soluble and can be manufactured by the body on exposure to UV radiation. They are also classified as derived lipids. You can get them when you expose yourself to UV radiation. That's why it's important that we get this early morning sun, all right, before 12 noon, for vitamin D to be synthesized, in our body. So exposure to sunlight will cause the synthesis of vitamin D. You can also get them from animal sources. You can also get vitamin D from animal sources. Now these are examples of calciferol. You have the egocalciferol, and then you have the cholecalciferol. All of them are steroid compounds, just like cholesterol. Now, functions of steroids. They are important components of cell membranes that alter membrane fluidity. By the time you get to 300 level, you'll be able to, when you are looking at the cell membrane, by the time you are studying cell membrane, you see the importance of cholesterol in cell membrane. They insert into the cell membrane and alter the fluidity of membrane. Membrane are supposed to be fluid. They are not supposed to be rigid. So when a membrane is rigid, this cholesterol insert and make sure that it makes it fluid. And when it is overly fluid, it also inserts and make sure that it is not too fluid. So that's why I said they kind of they alter membrane fluidity. They also function as signal molecules. The steroids play a vital role in cell signaling by helping regulating the process of development. So they play a vital role in signaling, and signaling will regulate the process of development, especially the steroid hormones. They regulate the process of development. In addition, they pass on messages received from outside the cell to effect changes inside the cell. These steroid hormones, they are synthesized from cholesterol, and what they do is they pass message from outside the cell to inside the cell to effect some changes, and the changes they are effecting could either be to regulate the process of development. All right, they can pass message to the nucleus where you have your DNA, and they regulate the protein synthesis. So they are very, very important signaling molecules. So by the time you get to 300, 400, you'll be able to see this particular function of hormones, all right, where they regulate the process of development. Now we've come to the end of classifying lipids. We've been able to classify lipids into 3 classes. Class 1 are the simple lipids, which are made up of the fatty fats and the waxes. I would say fats are esters of fatty acid and alcohol, and we said the alcohol could either be glycerol or a long chain monohydric alcohol in waxes. Then we said the second class are the complex or the compound lipids, and these complex or compound lipids are also divided into 3. We have the phospholipids, the glycolipids, and the lipoproteins. The phospholipids are also divided into 2: the glycerol phospholipid, which has glycerol as their alcohol, and also the sphingospholipids, which has sphingosine as their as their alcohol. and then we moved on to derived lipids, where we looked at the steroids, the cholesterol, we looked at fatty acid. Now we are moving forward to look at fatty acids. What are fatty acids? We have been talking about fats or lipids all along, but we want to look at fatty acids. What are fatty acids? Fatty acids are the building block of lipids. They are the building blocks of lipids. So lipids are made from fatty acids. Number 2, they are long unbranched chain carboxylic acid with linear unbranched carbon chain. So they are long unbranched chain carboxylic acid. The COOH at the end shows that it's a carboxylic acid. All right, they are linear. If they are unbranched, it means that they are linear, and they are carbon chain. They have carbon chain. Number 3, they are naturally occurring monocarboxylic acid which nearly all have an even number of carbon atoms. Most of them have even. There are some that have odd number of carbons, but majority of them have even number of carbons. We are talking about 4 carbon, 6 carbon, 8 carbon, 10 carbon, 12 carbon, 14, 16, 18, and so on and so forth. So they are all of the majority, not all, majority of them have even number of carbons. Then this fatty acid could either be saturated or unsaturated. It can either be saturated or unsaturated. You could either be saturated or unsaturated. Now, saturated fatty acid. What are saturated fatty acid? Saturated fatty acid, they have single bonds. Carbon-carbon bonds are single bonds. They also have even number of carbon atoms. Now, there are 3 types of saturated fatty acids. Number 1, we have the long chain fatty acids, saturated fatty acid, which can be carbon 12 to carbon. It can have a chain of carbon 12 to carbon 26. The chain length can be in between 12 and 26. And the 3 most important, or the most abundant, 3 of them that are most abundant are the palmitic acid, all right, and the stearic acid. Palmitic acid, stearic acid, and then I think we have the oleic acid. These are the 3 most important long chain fatty saturated fatty acids. Then we have the medium chain fatty acid, carbon 6 to carbon 11. Then we have short chain fatty acid, carbon 4 to carbon 5. Let's look at the numbering of the carbon atoms of fatty acid. The numbering starts from the carboxylic end group, the carboxylic group. So for example, let's look at lauric acid. Lauric acid has 12 carbon atoms and has no double bond. So the structural notation will be 12 colon 0. 12 means that lauric acid has 12 carbon but 0 double bond. It does not have any double bond. Okay. Now let's look at this example. These are saturated fatty acid and their structural formulas. Now this is caproic acid. Caproic acid has 6 carbon. Let's count it. Carbon 1, carbon 2. Now look at this. This CH2 is 4. So this is 1 plus 4, 5, 1 plus 1, 6. This is the carbon. So we said numbering starts from this end. Starts from the carboxylic end. So this will be carbon 1. This will be carbon 1. Then carbon 2, 3, 4, 5, and then carbon 6. The second one is caprylic acid. It has 8 carbon. If you count it, it's 8 carbon. If you have capric acid, 10 carbon. lauric acid 12, myristic acid, all right, myristic acid is 14, palmitic acid 16, then stearic acid 18, then arachidic acid 20, then lignoceric acid 24. So these occur as major fatty acids in human storage fat. So these are examples and the formulas for all these saturated fatty acids. Then the second type of fatty acids are the unsaturated fatty acids. The unsaturated fatty acid, they possess double bond. Instead of a single bond, they possess double bond, and they are classified as monounsaturated and polyunsaturated fatty acids. Now, monounsaturated fatty acids, they possess only one carbon double bond, carbon double bond. Okay, and the most abundant is oleic acid. It's oleic acid. 18 shows that it has 18 carbon and 1 double bond. 18 carbon and 1 double bond. The numbering of unsaturated fatty acid also starts from the end of the carboxylic end. For example, if you have 18, 2, it means the fatty acid has 18 carbons and 2 double bonds. Now let's take example of crotonic acid. Crotonic acid has 4 carbon, then 1 double bond, and this 2 is showing that it's at position 2. Now if you count it, 1, 2, 3, 4. So it's saying it's a 4 carbon, it has 1 double bond, then it is attached at position 2. Let's count. 1, 2. So it's attached to this carbon. Palmitoleic acid is 16, 1 and position 9. So if you count, this is 1 plus 7, that's 8 plus 1, 9. So it shows that it has 16 carbon, 1 double bond, and the position of attachment of the double bond is at carbon 9. Then oleic acid. Oleic acid is an 18 carbon. It also has 1 double bond. Double bond is attached at carbon 9. All right, so 1 plus 8 plus 7, 8 plus 1, that is 9. Okay, so it's attached at position 9. Now look at this omega 9. We'll get there, but look at it, omega 9. When you are counting, when you are talking of omega 9, you are counting from the methyl end here, not from the carboxylic end. So if you are talking of omega 9 fatty acid, you start counting from this end. This is carbon 1 plus 7, 8 plus 1, 9. So this is an omega 9 fatty acid. At the same time, it is also 18, 1, 9. If you count from this place, it's also 9 at position 9. It will be further explained. Let me explain it again. You start numbering from the carboxylic end of the fatty acid. So the carboxyl carbon here will be 1. Then you have 7 of this group, so making 8, then plus 1 making 9. So it shows that the double bond is attached at position 9. But when I'm talking of omega 9 fatty acid, you start counting from the methyl group. Carbon 1 plus 7 making 8 plus 1 making 9. So the double bond is on this carbon 9. So it's omega 9 fatty acid. Now for polyunsaturated fatty acids, we have more than one. We have 2 or more carbon-carbon double bond present. we can have up to 6 double bonds present in the fatty acid. So, and you must also note that in majority of these polyunsaturated fatty acid or unsaturated fatty acid generally, the cis isomer predominates. The trans isomers is rare. I hope you know what cis isomer means. So this cis configuration imparts lower melting points than their saturated counterparts, and the greater the degree of unsaturation, the lower the melting point. So the lower, the higher the degree of unsaturation, the lower the melting point. So when you have more of double bonds, you have low melting point for that fatty acid. So you have important polyunsaturated fatty acid, the omega-3 and the omega-6 fatty acid. They are very, very important polyunsaturated fatty acids. Now let's look at the structures of the polyunsaturated fatty acid. For example, we have the linoleic acid, alpha linoleic acid, ALA. It's an 18 carbon, and it has 3 double bonds, 1, 2, 3. Now this is called N3 or omega-3 fatty acid. It's called omega-3 because if you count from this methyl group, carbon 1, carbon 2, carbon 3, you can see that the double bond is attached to the 3rd carbon here. All right, but if you want to just name this compound and you are counting from the carboxylic end, 1, 2, 3, 4, 5, 6, 7, 8, 9. So it will be 18, 3. It could be delta 9. All right, 18, 3, 9. 3 means that it has 3 double bonds and it is attached at position 9. But if we are counting as an omega-3 fatty acid, we start counting from this methyl end. All right, and this will be carbon 1, carbon 2, and carbon 3. That's why they are called N3 polyunsaturated, that is omega-3 polyunsaturated fatty acid. So you have the alpha linoleic. Then this is another N6. This is omega-6 polyunsaturated fatty acid. Counting from the methyl end, it will be at position 6. This is 1, 2, 3, 4, 5, 6. Position 6. So that's why they are called omega-6 polyunsaturated fatty acid. So you can see it has 2 double bonds, but if you are counting from here, the double bond will be on 1, 2, 3, 4, 5, 6, 7, 8, 9. To be on 9. So these two, linoleic and arachidonic acid, they are an omega-6 polyunsaturated fatty acid. Now the cis configuration is what we have here. You can see them like they are facing down. That's the cis configuration. When it is trans, it doesn't look like this. It goes up. Okay, instead of coming down, it goes up. That's a trans form. But all these are in cis form. So you have the eicosapentaenoic acid and decosahexaenoic acid. All these are omega-3 polyunsaturated fatty acids. Now, the fatty acids are also classified as essential and non-essential fatty acids. The essential fatty acids means that they must be part of diet. It means that it cannot be synthesized by the body and they are necessary for proper metabolism. They cannot be synthesized in the body, but they are necessary for proper metabolism. That's why they are called essential fatty acid. And majorly the omega-6 and omega-3 fatty acids are referred to as essential fatty acids, EFA, the omega-3 and the omega-6 and omega-3. they cannot be synthesized, so they must be supplied by our diet, by what we eat from the food. It must be gotten from the food that we eat. That's why they are called essential fatty acid. We have seen the omega-6 series and the omega-3 series the other time. Linoleic acid is essential fatty acid from which gamma linolenic acid is derived. All right, and this gamma linolenic acid is primarily found in mother's milk, breast milk, and evening primrose seeds, and also the dihomogamma linoleic acid DGLA is also found in mother's milk, and also found in organ meats such as spleens, kidneys, and adrenals. So all these are omega-6 series, and they are essential fatty acid. They must be supplied in the body. That's why it's important for women to breastfeed their children, because it contains the essential fatty acid that is needed for growth. Also, these are the omega-3 series. The alpha linolenic acid is all these are the omega-3 series, which we have seen the other time. The non-essential fatty acids, they are various amino acids. They are required for normal health and growth, but they can be synthesized within the body or derived in the body from essential amino acids, so they are not that needed in the diet. This includes the arachidic acid, stearic acid, palmitic acid. All this can be synthesized by the body, although you can still get them from the food or the diet, but they can be synthesized. They are not needed to be included in the diet because they can be synthesized by the body. Now, the physical properties of fats. Water solubility. The short chain fatty acid, they have some sort of solubility, whereas the long chain fatty acids are insoluble. So the short chain fatty acid are sparingly soluble because of the carboxylic acid polar group. They can be little soluble because of the carboxylic acid, which is a polar group, but the long chain fatty acids are completely insoluble in water. So the longer the chain, the higher the higher the insolubility, and the shorter the chain, the more the solubility. The melting point. Melting point depends on the number of carbon atoms and degree of unsaturation. So as the melting point increases, as the number of carbon in their hydrocarbon chain increases. So the longer the chain, the higher the melting point. The longer the chain, the longer the fatty acid chain, the higher the melting point. Also, triglycerides that is rich in unsaturated fatty acids are generally liquid at room temperature and are called oils. So when the triglycerides, just like I showed you the other time, where you have 3 fatty acids are esterified to a glycerol. When you have the fatty acids, when the 3 of them are unsaturated, they are oil, they are liquid. They will be liquid at room temperature, and they are called oils. But when they are the fatty acid present are saturated fatty acid, they are generally semi-solid or solid at room temperature. That's why you have some, especially this cold season now, you'll find out that some oil in your kitchen might look solid or semi-solid, right, at room temperature, whereas some vegetable oils are liquid at room temperature. So those ones that are generally liquid at room temperature are called oils, and they are liquid at room temperature because they have more of unsaturated fatty acid. the triglyceride contains more of unsaturated fatty acid, and when they are solid, you know that they contain more of saturated fatty acid. So the more the length, the more the higher the melting point, and when the shorter the chain, the lower the melting point. So the effect of unsaturation on physical properties. Saturated fatty acids are well ordered, and they are compact crystalline forms. So they are well ordered, they are very compact, so they melt above room temperature, as exemplified by animal fats. So they are not oils at room temperature, they need a higher temperature to melt them. So the more unsaturation lowers melting point, saturation increases melting point. Let me say it again: unsaturation lowers melting point, and saturation increases melting point. So hydrocarbon chains, they have less ordered structure, they are not well ordered like saturated, because they have what we call kinks. There are kinks in their structures, and because of this, the triglycerides have melting points below room temperature. So unsaturated fatty acids have low melting point, whereas saturated fatty acids have high melting point. Now let's look at tests to check the purity of fats and oil. Number one is what we call acid number. Acid number is the number of milligrams of potassium hydroxide that is required to completely neutralize the free fatty acid that is present in 1 gram of the fat or oil. We call it fatty acid. So it is the acid number. You want to know the acid number. It's a fatty acid, it's an organic acid. So the number of milligram of potassium hydroxide that will neutralize the free fatty acid present in 1 gram of the oil or fat is known as acid number. So in normal circumstances, refined oils should be free from any free fatty acids. So oil on decomposition due to chemical or bacterial contamination yield free fatty acid. So what we are saying is that triacylglycerol are esterified, so they are not, the fatty acids are not supposed to be free. So but oil can decompose due to chemical or bacterial contamination, and when this happens, it shows that our oil is bad, that oil is not good. So when you have increased acid number in an oil, it shows that it's unsafe for human consumption. Increased acid number shows that it is unsafe for human consumption. So you use acid number to check whether the fat is good for human consumption or not, and increased acid number shows that the oil is not safe for human consumption. Then saponification number. Saponification number is defined as the milligram or the number of the potassium hydroxide required to hydrolyze or saponify 1 gram of fat or oil. So saponification number is a measure of the average molecular size. You want to see the kind of fatty acid that is present, average molecular size of the fatty acid present. So the value is higher for fats containing short chain fatty acids. So the saponification number will be high for short chain fatty acid. Majorly long chain fatty acid are the ones that are most suitable for consumption. So if it is high, then it shows that you have more short chain fatty acid. But if it is lower or is low, then you have a long chain fatty acid present in that fat. So it is a measure of the average molecular size of the fatty acid that is present. Then iodine number. Iodine number is defined as the grams or number of iodine that is absorbed by 100 grams of fat or oil, and it is generally used to know the unsaturation of fat. It's useful to know the relative unsaturation of fat, and it is directly proportional to the content of unsaturated fatty acids. So the lower the iodine number, the less is the degree of unsaturation. So it will help you to know, because unsaturated fatty acid is better for consumption. So when you have a low iodine number, it's showing you that you have more of saturated fatty acid present. But when you have high iodine number, it's telling you that you have a high degree of unsaturation present. So iodine number, determination of iodine number will help to know the degree of adulteration of a given oil, whether the oil is good for consumption or not. Like I said, if it is low, it might not really be good for consumption. The higher the iodine value, the better the fat or the oil. Now, chemical reactions of fatty acids. Number one, we have esterification. Esterification is a reaction between fatty acids with alcohols to form esters and water. So fatty acids react with alcohol to form esters and water. So these are fatty acids, all right, plus glycerol. Glycerol is the alcohol. These are the fatty acid glycerol, and you have triglyceride. So you can see this, the triglyceride are esters, or is an ester, and you have water being formed. For each esterification process here, you have one molecule of water released. So we have 3 molecules of water at the end of esterification between 3 fatty acids and glycerol. So the hydroxy groups of the carboxylic acid will pull the hydrogen proton from an alcohol to form a water molecule. From there, the resulting positive and negative molecules will attract each other and form a strong ester bond. So when you abstract hydrogen group from the fatty acid and hydrogen from the alcohol, what you will have is an ester bond. An ester bond is formed here, all right? It's an ester bond, all right? And then you have water being released. Is one of the reactions of fatty acid. Then we have hydrolysis, fatty acid hydrolysis. Hydrolysis reverses esterification. That is, fatty acids are produced from esters. For example, you have the ester, all right? Remember, this is our ester. Then you have water. Then what we have is fatty acid. It's a reversal of esterification and alcohol, just like we have between glycerol and the fatty acid. So what we have will be our fatty acid and alcohol. Instead of the ester that we had before, we can hydrolyze this so that we can get, you hydrolyze this, and then you add hydrogen back to the ester to form fatty acid, and then the alcohol is released. So you have fatty acid and alcohol. That is the end product of fatty acid hydrolysis. Then the other one is saponification. It's also one of the reactions of fatty acid. Ester reacts with a base to form a salt and an alcohol. The salt is normally known as our soap. It's known as soap. So sulfocation is the base catalyzed reaction hydrolysis of an ester. All right, and the products of the reactions are an alcohol. Okay, it's an alcohol, an ionized salt, which is a soap. So this is a salt of this is a salt which is a soap, and that's why it forms micelles that dissolve oil and dirt particles. That's why you use soap, you know, in washing your oil and dirty particles at home. So it's a reaction between ester, all right, and a base. Ester is your triglyceride plus a base, which is sodium hydroxide. Then you have a salt, and then you have an alcohol. Your salt is your soap. So rancidity. Rancidity is an oxidation reaction, all right, and the double bonds in triglycerols are subject to oxidation with oxygen in the air, all right, which is an oxidizing agent, and this leads to the breakage of the bond. So oxidation of these double bonds can lead to the production of aldehyde or carboxylic acid, which leads to production of odors which are not too good, especially in oil. That's why when you keep an oil for too long, at the end of the day you discover that it has become rancid. It produces an oil that comes as a result of an aldehyde or carboxylic acid being produced. So the oxidation process leads to spoilage of your fat or your oil at home. So to avoid this unwanted oxidation process, antioxidants are generally added as preservative. Antioxidants such as vitamin C and vitamin E are good preservatives to prevent oxidation of the double bonds, leading to production of aldehyde and carboxylic acid, which have objectionable odors that might not be too good, you know, and will lead to the fat becoming rancid, which is very, very unsuitable for human consumption. Now we have come to the end of the topic, structures, functions, and chemistry of lipids. If you have listened carefully, we've been able to look at define lipids, we've been able to say what a lipid is, look at how lipids are classified, looked at fatty acid, which is a building block of the fatty acid. We have seen how to number our fatty acids. We've been able to also see how to look at the reactions of fatty acids. So if you have any question for me, you can bring it up in class, or you can send it to me as an email. My email is titibamidele@unsuk.edu.ng. So thank you very much for your attention.